laptops. I don't want students having answers on their laptop, showing their friend right next to them answers to questions while they're taking the test. No. Take a nap. Again, I don't want you opening up a book, showing it to the person. Take a nap. Completing an SI review session, which is an excellent motivator to go to SI, which is awesome. I can't say enough good things about SI. Let's talk about blood vessels. So we already talked about blood. We talked about what blood is made out of. Cells, and it has a lot of plasma as liquid components. Blood is circulated by the heart. It's the double pump that moves the blood. Then the tubes that move the blood are the vessels. Multiple kinds of vessels. We have hepatic vessels, which we're not talking about in this chapter, but we have blood vessels as well. And as we look at blood vessels, I need to really emphasize this. Arteries away. Arteries. Arteries always carry blood. Usually 20%. Think that the pulmonary artery is going to carry heart because it has deoxygenated blood. So make sure we don't focus on the oxygenation state of blood. Focus on the direction of travel. Arteries are always going to go away. Veins are always going to go towards. And then when we look at those capillaries in between the arteries and veins, that's where the transfusion or the diffusion of nutrients is going to take place. In a typical blood vessel, we have some layers to the walls. These layers that wrap around the blood vessels are called tunics. When we think of the tunica or the tunics, we have an internal tunic or layer, we have a middle layer, and then we have an external tunic or layer. When we look at the tunica interna, the internal layer or tunic, this tunica interna is surface that's directly exposed to our blood vessels, or to the blood within the blood vessels. And because it's exposed directly to the blood, it needs to be very slippery, it needs to be very smooth. It cannot have a lot of friction. I mentioned this last week, but it's worth is identical to the endocardium, that inner lining of the heart. And that's because the inner lining of the heart also needs to be super smooth, super slippery to maximize the rate of blood flow. As we're looking at the endothelium that makes up the tunica interna, it's a simple squamous layer. Simple squamous, if you don't remember from last semester, is a one layer thick of cells. Simple meaning one layer. Squamous means flattened cells. So we have one layer of flattened cells that makes up the inner lining of our blood vessels. Because it's one layer of cells thick, it's going to maximize the rate of diffusion. We also have tunica media. Sometimes the tunica media is referred to as the muscular layer of the blood vessels. And that's because it has lots of smooth muscles that make it up. And as we look at different blood vessels will have different compositions to the tunica media. There's a lot of variation in the tunica media from blood vessel to blood vessel. Whether we're looking at large arteries or medium arteries or arteries versus veins. You may recall from last week when you went back and looked at the pig heart. Some of the blood vessels were sprung wide open, and they remained open with an exposed lumen. Other blood vessels had their lumens collapse in on themselves. And the reason that some blood vessels were sustained open was because they had a very thick, well-developed tunica media. Those other blood vessels that collapsed in on themselves had an underdeveloped tunica media. They were not supported and hence collapsed in on themselves. Some key differences between our tunica medias involve how much smooth muscle there is and how many elastic fibers there are to give it some recoil strength. So we can primarily vary the amount of smooth muscle and the amount of elastin or elastic fibers. And then finally, we have the outer layer of our blood vessels. The outer layer is referred to as the tunica externa. An old school name for it is the tunica adventitia. Occasionally when you're reading literature or you're looking in older textbooks or other textbooks, it'll just be called the adventitia. You won't even use the word tunica when referring to this structure. This outermost layer has a lot of connective tissues that serves to anchor our blood vessels in place. And in particular, for the larger blood vessels, so thinking like the aorta or the femoral vein or the external iliac artery, some of these really big blood vessels that we have, the tunica media is so thick. There's so many layers of smooth muscle cells making up that middle layer, and there's so many layers of tissues making up the outer layer, the tunica externa, that there needs to be a separate blood supply to supply the tissues of the blood vessels, to keep those tissues alive. And that separate blood supply that keeps the tissue of the blood vessels alive is called the vasa vasorum, or vasorum. And as we look at the vasa vasorum, it's just a secondary branching of blood vessels that goes into the outer walls of large diameter arteries and veins, and then it will branch off to form little capillaries in the large diameter walls and veins to supply nutrients to the tissues and remove waste products. As we look at the arteries, and recall, arteries always go away from the heart. If they are in the systemic circuit, those arteries are going to contain oxygenated blood. If they are in the pulmonary circuit, pulmonary arteries contain deoxygenated blood. And we refer to the arteries as resistance vessels because they have so much smooth muscle in their tunica media, and they have so many elastic fibers in their tunica media. The high concentration of smooth muscle and elastic fibers in the tunica media of the arteries allows for them to stretch out when we have ventricular systole. So when the ventricles are pushing blood out of the heart, our blood pressure spikes. We have our systolic blood pressure. The arteries stretch out and expand a little bit. And then when we have ventricular dystole, when we have the systolic blood pressure, the arteries squeeze back in on themselves. They recoil back to the resting length to help maintain baseline blood pressure. This elastic property of the arteries helps to maintain homeostasis for our blood pressure. Now, as we look at our arteries, the big ones, the really big ones, are going to be the aorta, common carotid, subclavian, pulmonary trunk, and common iliac arteries. These are enormous arteries that have a lot of secondary blood vessels supplying their tunica media. Something else that's key or very important about these arteries is that we can also call them the conducting arteries or the elastic arteries. We call them elastic arteries because they have lots of elastic tissues in their tunica media. Another name for them is conducting artery because they serve to transmit blood from one area to another area. These very large blood vessels don't actually deliver nutrients directly to the tissues of our bodies. That is the job of the blood capillaries. The purpose of the conducting arteries is to get the blood to the capillaries. The capillaries can have nutrient diffusion. We're also going to have distributing arteries and resistance arteries. When we look at distributing arteries, these are the arteries that go to individual organs. Most of the arteries that you're going to be learning in lab this week are either conducting arteries or distributing arteries. So we're going to learn the really large arteries of the body and then the smaller organ-specific arteries of the body. These distributing arteries are sometimes referred to as muscular arteries because they have a lot of myocardium. They have a lot of smooth muscle in their tunica media, but they don't necessarily have a lot of elastic fibers. The conducting arteries need a lot of elastic fibers in their tunica media because they are the closest to the left ventricle of the heart, and they're exposed to the highest pressure fluctuations during ventricular systole and ventricular dystole. And then finally, we have resistance arteries. These resistance arteries sometimes are referred to as arterioles. The relationship between an artery and an arterial is kind of like the relationship between an organ and an organelle. I've always thought of an organelle just as a teeny tiny organ inside of a cell. And when you think of an arteriole, you can think of it as a tiny artery, or just a small artery that is going to deliver blood directly to a capillary bed. So after our arterioles, we're going to go directly to the capillary bed, where we have nutrient diffusion from the blood. One kind of arterial that we can look at is the meta-arterial. And that meta-arterial is going to link for us arterioles to capillaries. So as we're looking at this figure that I included from your text, we have a meta-arterial that's going directly through a capillary bed. Leading to the arterial, we have another artery. Leading away from the arterial, we have a vein. Then branching of the meta-arterial, we have many tiny little openings for the blood capillaries. Something that's worth emphasizing with these meta-arterials is that when we're on the artery side of the meta-arterial, or the side of the meta-arterial that's closest to where blood is being delivered. So I can take my green marker here and just draw a line right down the middle. This is the arterial side. It's closer to the artery. The venous side is the half of the capillary bed that's closest to the vein, or it's going to be focused on removing the waste products. So on the arterial side of the capillary bed, we have a lot of little blood vessels, or excuse me, a lot of little muscles called sphincters. And these little tiny sphincters will constrict and cut off access to the capillary bed. In other words, we can control blood supply to our tissues. If we're in a situation where we need to deliver a lot of nutrients and remove a lot of waste products, we dilate or relax the sphincters and flood the capillary bed with excessive amounts of blood. If we're in a situation where we do not need a lot of nutrients, we constrict those capillary beds and reroute blood to other parts of the body where it's more important. You probably had some overprotective parents when you were younger that said something like, you can't go swimming within 20 minutes of eating food, or something of that nature. And the idea behind that is immediately after you eat a meal, the capillary beds of your digestive tract have dilated, you're sending blood to your digestive system, and you won't have as much blood going to the muscles of the legs and arms, which would make it more difficult to go swimming. And it's a primary reason why most of us don't like exercising immediately after a meal. Within our arteries, we need to monitor blood chemistry and blood physical characteristics. Our blood and our cardiovascular is a key component for maintaining homeostasis in our body, or that relatively constant internal environment. So some of the key factors: the chemistry of our blood and our blood pressure. And we use different kinds of sense organs to monitor these things. We covered this last semester, but it's worth reiterating because it's been a while. We have baroreceptors, and when we think of baroreceptors, look at the word baro. It's the same word in a barometer, and it's used to measure the pressure. So we measure blood pressure with a baroreceptor, and then we measure blood chemistry with chemoreceptors. As we look at baroreceptors, the only thing they're interested in is the pressure. When we look at blood chemistry, there's multiple variations of the chemoreceptors. Some look at hydrogen ion concentration, or pH. Some are designed to measure oxygen concentration, and some are designed to measure carbon dioxide concentration. These are the three variables that we're most interested with in regards to blood physiology and homeostasis. Now, when we look at the location of these sensors, there's some common spots in the body where we can find them. In particular, right at the ascending aorta and aortic arch, we are going to monitor blood pressure. We have a lot of baroreceptors located at the point where the blood is directly leaving the left ventricle, where we have the absolute highest blood pressure in our bodies, or I should say within the systemic circuit. We're also going to have some chemoreceptors located as we send blood up to the brain. We have chemoreceptors directly monitoring the blood that goes to the brain, because our brain is the single most important organ in our body and is very concerned with maintaining proper oxygen input, proper sugar input, and the proper pH. And then finally, we also have some baroreceptors leading in the blood vessel, or in the blood vessels that lead to the brain. That particular baroreceptor in the carotid sinus is going to sense the change in blood pressure when you go from lying down to standing up. And as it senses that change in blood pressure, it's going to cause the heart to have higher contraction strength. It will increase cardiac output, and it will also trigger the smooth muscle of the blood vessels to constrict, to elevate our blood pressure, to help keep you from getting dizzy and passing out when you stand up quickly. So when we talked about arteries, they were resistance vessels because they had to resist fluctuations in blood pressures, or blood pressure changes. When we think of capillaries, we can think of them as the exchange vessels. These capillaries allow for us to exchange nutrients and waste products and fluids, or just chemicals in general. Generally speaking, we'll say that they are used to deliver needed nutrients, and they're used to remove excessive waste products from the tissues of our bodies. A common student misconception is that the blood capillaries are going to deliver the nutrients directly to the tissue, directly to the cytoplasm of the cell in the tissue. And that is an incorrect assumption. When blood capillaries are delivering nutrients, they deliver the nutrients to the extracellular fluid, and from the extracellular fluid, those nutrients will then diffuse into the cell across the cell membrane. So many students forget about that extracellular fluid. That's a key component as we're looking at the exchange of nutrients. There are some tissues in our body that don't have capillaries for various reasons. A classic example is the lens of your eye. And the reason we don't have any blood capillaries in the lens of our eye is because those capillaries would block the transmission of light. We don't want to block light from going through our lens. Another classic example of a tissue that has very low vasculature is going to be all of the epithelial tissues. We have very low amounts of vasculature in those epithelial tissues. So the tendons, ligaments, so on and so forth. Think of stratified squamous of our epidermis, or you can think of the tendons and or ligaments of our skeletal system or muscular system. These tissues that heal very slowly when they're damaged have very low amounts or almost no blood capillary supplies. As we look at a blood capillary, it has, and I'm going to emphasize this here, not much to it. It has an endothelium on the inside of that blood capillary, and then it has some connective tissues on the surface of the blood capillary to help anchor it into place. Blood capillaries do not have smooth muscle present. The reason blood capillaries do not have any smooth muscle present within their wall is because that smooth muscle would slow down the rate of diffusion. We want to get the nutrients across the membrane as quickly as possible, so we make the membrane as thin as possible with our blood capillaries. And there's variations of blood capillaries in our body, depending on where they are in our body and the particular function of that capillary. The most common kind of capillary in our body is called a continuous capillary. It's the simplest, most straightforward capillary that we have. And as we look at this continuous capillary, its endothelial cells are going to have lots of tight junctions in it. The tight junctions will hold the endothelial cells together so that we have a water connection. Another way of thinking of this is those tight junctions keep water from moving in between the cells. Instead, when something is going to leave the capillary, it's going to need to go through the cell directly. So as we're looking at the transmission of materials, we're going to have a lot of materials going directly through the cell. The regions with tight junctions, in areas with no tight junctions. And this area with no tight junctions within the endothelial capillary is going to be open to allow liquids to transmit across the tight junction, or through the cleft or through the opening. As we look at our other two capillary types, the fenestrated and sinusoids capillaries, these ones are not that common in our bodies. When we look at a fenestrated capillary, I want you to think of a capillary that's very leaky. A fenestrated capillary has lots of holes in the endothelium. And this extreme leakiness of the fenestrated capillary is going to allow for liquids to transmit very quickly across its barrier. And we find lots of fenestrated capillaries within organs that specialize in rapid movement of fluids from the blood to the extracellular fluid or to other fluid beds within our bodies. In particular, we have a lot of fenestrated capillaries in the kidneys and the small intestines. We haven't talked about either of those organs yet. We haven't gone to the urinary or digestive systems. When we think of kidneys, we have the rapid diffusion of blood plasma across the capillary as we are filtering the blood to produce urine. And then within our small intestine, we need to have the rapid absorption of nutrients from the lumen of the small intestine to our blood capillaries. We also have sinusoids capillaries, or discontinuous capillaries. These capillaries are the rarest capillaries in the body. And the reason they're so rare is because of their function. They have extremely large openings in them. And these extremely large fenestrations, or openings, are going to allow for large molecules, like proteins, to enter the blood. Or we're going to allow for red blood cells to enter the bloodstream through those extremely large openings. The blood cells need to get into the blood somewhere, and it's going to be in the sinusoids capillaries. We're going to have these capillaries present in tissues that specialize in processing or producing red blood cells, or that specialize in regulating blood protein content. So when we think of the liver produces a lot of the blood proteins or serum proteins, so it's going to have some sinusoids capillaries, but we're also going to have those capillaries present in the red bone marrow, so new blood cells can be added to circulation. We have this kind of capillary present at the spleen, so we can remove damaged, broken, or degraded red blood cells from circulation. We also have present in our bodies veins. The arteries always go away from the heart. Veins always go towards the heart. They're named based off of directional flow. And we call our veins the capacitance vessels. Arteries are resistance vessels that help to regulate blood pressure. Our capillaries are the exchange vessels. We will exchange nutrients between the blood and the tissues of the capillaries. And then for the veins, they're capacitance vessels. Most of our blood capacity is going to be in our veins. Most of the blood of our body is stored in the veins. If we were to look at the systemic circuit, 64% of the blood of our body, or the vast majority of the blood of the systemic circuit, is going to be present in our veins of the systemic circuit. The reason why we can store so much blood in our veins is because they have a very wide diameter lumen. The opening on the inside of our veins is much wider than the opening on the inside of our arteries. And as we're looking at our veins, the wall of our veins is going to be significantly different compared to the walls of our arteries. Arteries have lots of smooth muscle and have lots of elastic fibers within the smooth muscle of the tunica media. As we look at veins, veins have almost no elastic fibers in their walls, and they have a lot less smooth muscle present in their wall. When I would work with cadavers with my students, key thing I would have them do is to look at the common iliac artery versus the common iliac vein. And when you're looking at them in a cadaver, they're all the same kind of bleh color, kind of buff color. And the only way you can really reliably tell them apart is to poke them with your finger. If it springs back, it's the common iliac artery. If it remains as a limp noodle, it's the common iliac vein. As we're looking at the way we can move blood through our bodies, pathways can be broken up into some common categories. The most common pathway is known as the simple pathway or simplest pathway. This is one that hopefully you knew before you started this class. Most high school biology classes cover the idea that blood will leave the heart, go through an artery, enter the capillary bed, exchange nutrients, and then be back to the heart. That's the simple pathway. And this simple pathway is by far the most common pathway in our bodies. We're going to have, and this is a key point, only one capillary bed for a simple pathway. It's also worth emphasizing that all the blood goes through the capillary bed. Other pathways or other circulation pathways either will have multiple capillary beds or will have bypasses that bypass the capillary bed, such as the portal systems. The most common portal system is the hepatic portal system, or portal system that carries blood from the small intestine to the liver, and then from the liver the blood is going to be sent to the heart. When we look at a portal system, its key or defining characteristic is the presence of two capillary beds. So we'll leave the heart through a conducting artery, and then we'll go to a transmission artery, to the arterial, to the capillary bed, to another arterial, to another capillary bed, to a venule, and then to a vein, back to the heart. A lot of students would often ask me, what's the point of a portal system? Why would we want multiple capillary beds in between the heart, or in between the left ventricle and the right atrium of the heart? And in particular with the hepatic portal system, it has to do with processing the food that we eat before we release those nutrients to the rest of the body. We eat a lot of stuff that's not necessarily the best for us to consume. In Wisconsin, we have a lot of alcoholism. And when we think of alcoholism, the organ that's most severely affected by alcoholism is the liver. When people die of alcoholism, they oftentimes will have cirrhosis of the liver, or where the liver in their body is breaking down. And the reason for that is when you consume the alcohol, it goes to your stomach, then to your small intestine, and then all those nutrients absorbed from the small intestine, either the good nutrients or the bad nutrients, all nutrients are going to be sent to the liver, where the liver will detoxify and process as much of the bad stuff as possible. And then after the liver detoxifies the stuff you absorb in the small intestine, it's then going to send the blood back to the heart and then distribute, hopefully, just the good nutrients to the rest of your body. When we look at our circulatory routes, we can have an anastomosis. When we think of an anastomosis, it's going to be blood vessels converging. And it's any convergence point where multiple blood vessels join together outside of the capillaries. So you can have arterioles joining together. You can have venules joining together. It's the merger of blood vessels other than capillaries. So when we think of an arterial or an arteriovenous anastomosis, this anastomosis is going to take an arterial and connect it directly to a venule. This will allow for blood. Depending on our tissue needs, we can reroute the blood directly past the capillary bed. This oftentimes is going to happen as we're having temperature fluctuations in our body. When we're in cold environments, we need to conserve body heat. We reroute blood past the superficial capillary beds, reroute as much blood as possible towards the core of our body to maintain our core body temperature. And then in situations when we are overheating, we will constrict sphincters to the anastomoses and reroute the blood towards the superficial capillary beds so that our surface tissues become flush with blood and we can radiate off as much heat as possible. Now, when we look at blood pressures in our bodies, guys, gals, our blood pressure is going to be directly connected to the left ventricle, the contraction and relaxation of the left ventricle. If you think about it physically, it's going to be the blood pushing against the walls of the blood vessel that pressurizes that blood vessel. When the left ventricle is contracting, it's going to increase the force of blood leaving the heart. It's going to pressurize the blood and cause systemic blood pressure to go up. The name of the device that measures blood pressure is a sphygmomanometer. And it's kind of a tongue twister. The technical name of the device that measures blood pressure is a sphygmomanometer. Most people struggle with saying sphygmomanometer and will instead just say blood pressure cuff. It's a lot easier to pronounce. But as aspiring healthcare professionals, you should know the technical name of the instrumentation you're working with. Now, there's two different blood pressures that we measure. We measure a blood pressure that corresponds to the contraction of the left ventricle and the relaxation of the left ventricle. Those are going to be the systolic and diastolic blood pressures. Since systolic pressure corresponds to contraction of our left ventricle, it's the big number. And diastolic number is going to correspond to the relaxation of the left ventricle, so it's the smaller number. When we think of the units, and units are very important, the units that we use are millimeters of mercury. The initial barometers, the very first barometers, had columns of mercury suspended in a vacuum against gravity. And it was found that regular atmospheric pressure at sea level was 760 millimeters of mercury. So when we think of our blood pressure, our systolic blood pressure is about 1/6 higher than regular atmospheric pressure. So these numbers are raw numbers above and beyond atmospheric pressure. So the 120 over 75 means that our blood is pressurized just a little bit above atmospheric pressure. So if we were to cut an artery in either yourself or in a patient, that 120 millimeters of mercury would be a pressure above atmospheric pressure, and the blood from the artery would squirt up into the air against the atmosphere. Now, if we were to look at venous return or venous supplies, the blood pressure in our veins is very low. The blood pressure in our veins doesn't fluctuate like it does in our arteries. So when there's damage to a vein, instead of having the squirts of blood going up against gravity, we just have a gentle oozing of blood that occurs. If somebody's blood pressure is above 140 over 90, they are officially at the threshold for hypertension. If they're above 120 for their systolic blood pressure, they're pre-hypertensive. They're not quite to the threshold of 140 over 90, but it's not good to have that elevated blood pressure. When we think of chronic high blood pressure or chronic hypertension, it's going to cause a lot of damage to the blood vessels. It's kind of like overinflating a balloon. If you overinflate a balloon and stretch it out too much, eventually the balloon loses the ability to spring back to its original shape. And if we overinflate our blood vessels with excessively high blood pressure, our arteries lose the springiness of their elastic fibers. We start to lose the ability to maintain a relatively constant blood pressure with our arteries. As our arteries stretch out and weaken, we can have aneurysms where they are going to burst spontaneously, or we can have an atherosclerosis form, which is going to be a plaque building up within the tunica intima of that blood vessel, or lipids building up in between the tunica media and the tunica intima. The opposite of high blood pressure, the opposite of hypertension, is hypotension, low blood pressure. Individuals that have chronically low blood pressure are prone to fainting a lot, and my wife falls into this category. Their blood is not pressurized, so they either have low blood volumes, or they have low contractile strength of the left ventricle, or they have an impaired ability to contract the smooth muscle of their arteries. And if they have one of those variables causing their blood pressure to be lowered, they will have a tough time pumping blood against gravity up to the brain to keep their brain properly oxygenated, hence the constant fainting. Other causes of low blood pressure could include dehydration. If we don't have enough water in our blood, we won't have enough fluid to fill our blood vessels. Or we could have lack of hemoglobin. So if we don't have enough hemoglobin, it's difficult to produce our blood. Or the common one is you could just And if you're constantly bleeding, you might not have enough blood to pressurize your blood vessels. Now, when we look at this blood pressure, we're going to change blood pressure primarily through cardiac output. So think stroke volume or heart rate, either by making more blood or changing the diameter of our blood vessels, by constricting the smooth muscles of our blood vessels. When we think of blood volume, most of the liquid that leaves our bloodstream is going to leave through the kidneys. So when we look at our kidneys, our rate of urination, how much urine we produce, is directly correlated to our blood pressure. Individuals that have chronically high pressure are prone to urinating more. Individuals with chronically low blood pressure typically will not urinate as much as their high blood pressure counterparts. So let's shift gears for a moment and give you a chance to apply what we've been learning. Never mind, we're skipping that question. I put that in the wrong spot. Sorry about that. We have not covered the answer to that question yet. We'll come back to that after we cover the hormones. So let's look at peripheral resistance of our blood vessels, guys, gals. As we're looking at our blood vessels, peripheral resistance is going to be the force that impairs or goes against the flow of blood. Our left ventricle wants to push blood out of the heart. Peripheral resistance wants to push blood back into the heart. We have the aortic valve or aortic semilunar valve. That aortic valve is going to help reduce the effect of peripheral resistance. It'll keep blood, hopefully, if it's functioning correctly, from flowing backwards into the left ventricle during ventricular relaxation or ventricular dystole. Some key factors that influence resistance include the viscosity, or how thick our blood is. If we have an accelerated or elevated amount of red blood cells or erythrocytes, if your hematocrit is at 66 to 70, you have an incredibly high ratio of red blood cells relative to the amount of blood plasma. Your blood will be extra viscous, and your heart has to pump extra hard to compensate for that. On the other end of the spectrum, if your blood hematocrit is at 20 or 30% red blood cells, you have hardly any erythrocytes. Your blood is going to be extra fluid. It'll be extra runny. The technical term is serous. It'll be extra serous, and it'll flow very easily because it has an increased flow rate. Another thing that influences resistance is the length of the tube. I remember when I was younger, my parents had a 50-foot garden hose. And sometimes when I was thirsty, I would try to suck water up the garden hose, even if the garden hose wasn't being turned on. And sucking water up a straw that is 50 feet long is kind of hard to do compared to a regular sippy straw you get at a fast food joint. The length of the tube is going to influence how hard it is to move something through the tube. When we're looking at a liquid and it's flowing through a tube, the liquid rubs against the side of the tube. And that fluid friction against the side of the tube is going to directly influence the amount of resistance we incur. Another one is vessel radius. And really just to simplify it, the wider the tube, the more liquid you can move. We can connect this to inner tubing. If you go inner tubing down the Chippewa River, you probably noticed that if you want to catch up to your friends, you should kind of paddle over to the middle of the river where the water is flowing really fast. And if you're ahead of the pack and you want to slow down, you paddle over to the edge by the stream bank where the water is not flowing as fast. And that has to do with the resistance from friction. Near the edge of the blood vessels, we have a lot of fluid friction. But in the center of the blood vessel, where the blood is not rubbing against anything, there's less fluid friction, and the fluid can flow faster. The wider the blood vessel, the less fluid friction there is in the center of the blood vessel. Another reason for this has to go back to geometry. Think back to either your freshman or sophomore geometry class in high school. We have the radius of a circle, pi r squared. And as we're looking at this, pi r squared, formula, the radius squared is going to influence the cross-sectional area of the circle. And because of some other factors at play, we're not putting the full equation up on the board right now, radius to the fourth power is ultimately going to influence our blood vessels. And what we end up seeing if we plug the numbers into the equation, which I'm not going to ask you to do for lecture, which is why I'm not giving you the equation for lecture, is that a small change in radius is amplified greatly because it's radius to the fourth power in the equation that regulates or explains the flow of blood. As we're looking at peripheral resistance in our bodies, another factor that influences our peripheral resistance is going to be the distance from the heart. And more specifically, the distance from the left ventricle. Because when we look at the superior vena cava or the inferior vena cava, those are pretty close to the heart, but they're really far from the left ventricle. We had to go through the entire systemic circuit, and when we're at those large veins that are dumping blood back into the heart, that blood has slowed down dramatically. If I back up a couple slides here, when we get to those vena cava, the superior and inferior vena cava, blood flow, here we go, is very slow. Part of that is the great distance the blood has traveled. The farther the liquid has to flow, the slower it's going to move. Another reason for that is within the arterial capillaries, we have a very narrow radius. The very narrow radius of the arterioles and capillaries causes a dramatic increase in fluid friction. The dramatic increase in fluid friction in capillary beds is going to slow down the rate of flow and going to increase the resistance. And then finally, if we look at cross-sectional area, the cross-sectional area, if we took all the diameter, all the lumens of all the blood vessels, the farther away from the heart we go, the larger the cross-sectional area will be. Another way of thinking of this is the more blood we can store. As we take the rate of flow and alter the cross-sectional area but keep the rate of flow the same, we're going to alter the speed. You've probably seen this with the garden hose. If you turn on your garden hose and just push water out of it, it usually has like a flow rate of about 3 to 4 gallons a minute. It's a typical household garden hose. But if you take your thumb and put it on the end of the garden hose, the same amount of liquid keeps coming out. You've changed the radius, and it will squirt out far into the air, so you can spray your siblings with it. The same is true with our veins versus our arteries. The overall diameters, the overall amount of cross-sectional areas of our arteries is much smaller than the cross-sectional areas of all the veins combined. Now, to regulate blood pressure, we have local control, neuronal control, and hormonal control. And as we're looking at these ways of regulating blood pressure, it's going to alter the movement of our vasculatures, the movement of blood through our blood vessels. Motion through our blood vessels. And we can regulate it at the tissue directly, we can regulate it with our nervous system, or we can regulate it with our endocrine system. Let's talk about some of the auto-regulations that we'll have at the tissue itself. Here's some methods of local control directly at the capillary bed. We can constrict and relax sphincters that control the capillary bed. If we look at a capillary bed, a lot of capillary beds will have meta-arterial going directly through it. If we relax the precapillary sphincters on the arterial side of the meta-arterial or capillary bed, we allow more blood to enter into the capillary bed, and that's going to increase vasomotion. We're also going to find that the smooth muscle within the blood vessel can dilate and contract. So when we look at vasodilation, this vasodilation is going to control the diameter or the radius of our blood vessels. During vasodilation, the radius will increase, and we increase flow rate and increase blood supply. And because radius to the fourth power influences flow rate, a very small change in the diameter of our blood vessels will have a very profound effect on the overall rate of flow. We also have some chemicals that influence local control. These vasoactive chemicals can cause an increase or a decrease in blood flow depending on our needs. So when we think of the basophils, the rarest kind of granulocyte, the basophils are going to release histamine, and histamine is going to cause dilation, or is going to cause the blood vessels to dilate. This is a key component of the inflammatory response. We have other chemicals at play, such as bradykinin or some prostaglandins. All of these are going to stimulate vasodilation. For those of you who don't remember, prostaglandins are a kind of hormonal transmitter that are based off of fatty acids. It's a fatty acid-based hormone. And then when we look at bradykinin, that is a protein-based signaling molecule. And then we also are going to have some other vasodilators that are going to be potentially inorganic, such as nitric oxide. Nitric oxide is a well-established vasodilator. And it's of people with high blood pressure taking a nitroglycerin tablet. The idea behind this is if you take nitroglycerin tablets, you upregulate the amount of nitric oxide in your body, and by increasing nitric oxide concentrations, you cause blood vessels to dilate and compensate for your chronically high blood pressure. Now, I don't know how much chemistry background you have, but when somebody says nitroglycerin, I usually get kind of excited by that because it has this tendency to explode spontaneously when you tap it with your finger. If you've ever heard the story behind Alfred Nobel and the Nobel Prize, he came up with a way to stabilize nitroglycerin by taking the nitroglycerin, putting it in clay, and then making dynamite out of it. And from a clinical perspective, patients don't take pure nitroglycerin tablets. They would explode on the car ride home. Or they explode in your hand as you're jiggling it in your pill bottle. Instead, what they're really taking, more accurately, would be dynamite tablets. The nitroglycerin has been stabilized with clay, and then you pop it in your mouth as a pill to help lower your blood pressure. I always found when we think of the prostacyclin is another signaling molecule that can be released to cause that vasodilation and the lowering of our blood pressures. Another example of local control is going to be reactive hyperemia. When we think of hyperemia, I want you to think of an increased supply or an increase of oxygenation. Anemia is reduced blood oxygen levels. Hyperemia is increased blood oxygen levels. We have reactive hyperemia when we need to restore the tissue back to homeostatic norms. So think of those times maybe you had a ring on your finger and it was too tight, or maybe you had a mild tourniquet on your body. I know it was common on the school bus when I was younger. We would wrap hands around our fingers to see how purple we can make our fingertips turn. And the idea behind this is after you cut off or reduce blood supply for a while, your tissues have lots of waste products built up. And then when you restore blood supply, so when me and the other idiot kids in the bus would cut the rubber band off our fingers, we would restore blood supply. We'd have an excessive amount of waste products in that tissue to upregulate blood supply, blood flow, to quickly remove the excessive amount of waste products that are built up. So it's a reactive hyperemia. We elevate blood oxygenation or blood flow to the tissue in response to a cut-off blood supply. We can also have angiogenesis. Genesis is a word that means to create. It's being phased out right now, but we're still using the term angiogenesis in this phase, or at least for this particular aspect of creation in our body. Angiogenesis is going to refer to the creation of new blood vessels. So we can have new blood vessels grow in a variety of situations. We grow new blood vessels in cancerous tumors. new blood vessels when we exercise a lot. If you're constantly exercising, your skeletal muscles are starved for oxygen and will grow new blood capillaries to those skeletal muscle tissues. During the menstrual cycle, the endometrium, the lining of the uterus, will grow new blood vessels once a month and then degrade those blood vessels once a month if fertilization does not occur. There are many aspects in our body where we can grow new blood vessels. Some areas of anti-cancer research right now are looking at the inhibitors of angiogenesis. If we can inhibit the growth of blood vessels to the cancerous tissues, we can starve the cancerous tissue of nutrients and make it so that the tumor does not grow. Let's talk neuronal control. When we think neuronal control, this is going to be associated with our central nervous system, and it's going to involve muscle or nervous action potentials controlling the dilation of smooth muscles in the tunica. It's also going to control the rate of contraction of our cardiomyocytes, or the contraction force of contraction of our heart. One of the neuronal controls at play is called the BIOL. We've talked about it in general, but let's really put it to writing. This is the idea that we need to maintain we have reduced blood pressure. In response to reduced blood pressure, in response to elevated blood pressure, our arteries are stretched out because of that high blood pressure. When we have high blood pressure, our blood pressure sensors, the baroreceptors, pick that up, and they cause two key things to happen. First, we are going to activate the vasomotor center of our medulla oblongata to help regulate the constriction or the tone of our blood vessels. In other words, we contract smooth muscle of the tunica media. And the smooth muscle of the tunica media. This is going to ultimately cause dilation and reduce blood pressure. And then when we look at the effects on the heart, as we're affecting the heart with our baroreflex, or baroflex as some people refer to it as, if we have high blood pressure, we need to reduce cardiac output. And we will reduce cardiac output by reducing the overall heart rate. We will increase vagal tone. As we talked about last week, the vagus nerve releases acetylcholine. That acetylcholine is going to affect the cholinergic receptors on the heart and reduce the overall heart rate. Ultimately, these two separate factors, the reduction in cardiac output and the dilation of our blood vessels, will work in conjunction to lower blood pressure back to homeostasis. However, this is short term. And when we think short term, I want you to think minutes, maybe hours, potentially hours, but very short term. This is not going to be a weeks or month mechanism for regulating our blood pressure. Longer term mechanisms are going to involve changing the fluid balance of our bodies or going to involve changing the protein content of the blood serum. We're also going to have some chemoreflexes associated with neuronal control. Depending on our blood carbon dioxide concentrations, our blood pH, and our blood CO2 concentrations, we are going to alter our blood flow in response to the changes in our blood chemistry. In this concept, when we say respiration right here, I am not referring to cellular respiration. Last semester, respiration referred to the generation of ATP at the cell. In this context, respiration refers to breathing in and breathing out. It controls how quickly you breathe in and out for this respiration that we're talking about. But we breathe in and out more, and I'm sorry that this cut off on us here. Breathing in and out more is going to alter the pressure in our thoracic cavity. During inhalation, when we're breathing in, we have lower pressure in our thoracic cavity, and that reduced pressure in our thoracic cavity is going to reduce overall blood pressure in our bodies. It's also going to change the amount of oxygen exchange between the blood and the gases in our lungs, and will also influence the amount of carbon dioxide exchange between the blood and the gases in our lungs. And by regulating those blood gases through respiration, we can influence our blood chemistry. We also are going to have some hormones that can influence, or hormones that influence blood pressure. These hormones can either upregulate, they can either increase blood pressure, or they can decrease blood pressure. For example, we have angiotensin II. Angiotensin II is a vasoconstrictor. As a vasoconstrictor, it causes blood vessels to have a reduced radius and is going to cause our blood pressure to go up. Another thing that angiotensin II does is it causes our kidneys to reabsorb sodium ions. And if we reabsorb more sodium ions into our bloodstream from our urine production, we're going to cause more water to be sucked into our blood via osmosis. So really, angiotensin II does two things to increase our blood pressure. It causes blood volume to go up through the reabsorption of sodium in our kidneys, and it causes blood vessel radius to go down through vasoconstriction. Both will separately raise blood pressure, and in conjunction will raise blood pressure dramatically. We also have atrial natriuretic peptide, or ANP. ANP is going to cause more sodium to be in the urine. And if we urinate out more sodium, that gives us less sodium in our blood. The less sodium ions in our blood, the less water we are going to store in our blood because the blood osmolarity has been reduced. So practically speaking, ANP causes us to urinate more. And as we urinate more, blood volume goes down, blood pressure goes down. Also known as vasopressin, antidiuretic hormone, or ADH, it causes our kidneys to reabsorb water directly during urine formation. A lot of students get ADH and angiotensin mixed up. When we think of angiotensin, think of sodium reabsorption. When we think of ADH, the antidiuretic hormone causes the water to directly be reabsorbed, as opposed to indirectly reabsorbed. Now, if we have very high amounts of antidiuretic hormone in our bodies, we are going to cause our blood volume levels to go up and blood pressures to go up. We also are going to have epinephrine and norepinephrine, also known as adrenaline and noradrenaline. These aren't going to influence fluid volumes, but they're going to influence cardiac output and blood vessel radius. They're both going to influence the blood vessels by causing vasoconstriction. In particular, we have alpha-androgenic receptors in our blood vessels. So the smooth muscle in our blood vessel constricts when exposed to adrenaline. On the other hand, we have a different kind of androgenic receptor in our cardiomyocytes. And as we look at those cardiomyocytes, we can have some beta-androgenic receptors in the blood vessels of our heart that dilate. So our cardiomyocytes have increased blood supply in response to adrenaline. And then the conducting vessels, those large arteries in our bodies, will have their smooth muscle constrict in response to adrenaline. In other words, the heart will pump harder and we will move blood to the capillary beds quicker in response to adrenaline and noradrenaline. Let's see how we're doing time here. We have 14 minutes left. So, guys, gals, let's talk about how we can use our vasoreflexes to help maintain homeostasis. As we look at these vasoreflexes, the goal is to maintain a relatively constant internal environment. When we think of homeostasis, it's the unifying theme in anatomy and physiology. So relative to our blood supply, we're going to need to maintain a relatively constant blood pressure, and we're going to need to maintain relatively constant blood chemistry. And we'll do that by rerouting blood to different tissue types and by changing vessel diameter or vessel radius. Now, if we want to increase our blood pressure, we will first need a baroreceptor to pick up the low blood pressure. And then that baroreceptor will send the signal to a part of the brain known as the medullary vasomotor center. Our medullary vasomotor center is in the medulla oblongata, hence the namesake of medullary vasomotor center. So it's a part of the brain in the brainstem, or medulla oblongata, specifically of the brainstem, that helps to control the hormones that regulate blood supply. This individual right now, he's resting, he has parasympathetic activation, and most of his blood is being rerouted to his intestinal tract. On our next slide, we have some individuals that are going through some athletic competition. They're using their skeletal muscles a lot, and these individuals are down-regulating intestinal blood supplies and up-regulating skeletal muscle blood supplies. This is controlled through both sympathetic activation of our body's nervous system, and it's also going to be controlled through the constriction of those pre-capillary sphincters on the arterial side of the capillary bed. We also find that during this sympathetic activation, we reduce the amount of blood flow to the kidneys. When we're relaxing, so for most of us in the room right now, we're working physically very hard, about 20% of the blood that leaves our heart is being routed through our kidney. And during intense exercise, the amount of blood going through our kidneys is reduced dramatically. One of the key things that will influence this rerouting of blood is the local control of blood supply. These are muscle tissues, our skeletal muscle tissues. We have a lack of needed nutrients. We have a buildup of waste products, and change in the tissue chemistry so that we can help to maintain homeostasis. As we look at our capillaries are going to exchange blood or exchange liquid with the extracellular fluid. And it's going to be a two-way movement of fluids. We'll have fluid enter the extracellular tissues or extracellular cavity, and then we'll have fluid leave the extracellular cavity and go back into the blood supply. This two-way movement of fluids allows for all of these nutrients and waste products that are listed here to go back and forth between the blood plasma and the extracellular fluid. Some chemicals can go directly through the blood capillary wall, and that's going to be through diffusion and transcytosis. Some larger chemicals can't fit directly through the blood capillary wall. They can go through the intracellular clefts, and then some very large things, such as blood cells, will have to go through some filtration pores. Of the ways to move things through our blood capillaries, diffusion is the single most important method. It's going to allow for very small molecules or for nonpolar molecules to move with their concentration gradients. So when we think oxygen gas and carbon dioxide gas, those are two key substances that can diffuse directly across the cell membranes very rapidly. If we think of the cell membrane, it's made of a phospholipid bilayer, and any lipid-soluble chemical compound can diffuse directly through the endothelial cells of the blood capillaries very rapidly as well. When we think of larger molecules, like a glucose molecule, which is mildly polarized, or a water molecule, which is also mildly polarized, these polarized or mildly polarized molecules have a hard time going directly through the phospholipid bilayer, and instead need to go through filtration pores. Extremely large molecules, like protein molecules, or large polymers, say of fibrin, for example, can't diffuse through the cell membrane at all, and need to have a different mechanism to get across. So when we think of diffusion, guys, gals, I want you to think of going directly through the cell that makes up the endothelium. Another method to get large molecules across the barrier is going to be transcytosis. Transcytosis is endocytosis paired with exocytosis. We'll have endocytosis where part of the cell membrane swallows or invaginates some chemicals. That transport vesicle moves across the cytoplasm, and on the extracellular fluid side of the blood of the cell, we'll have exocytosis, dump the contents into the extracellular cavity. So when we think of transcytosis, I want you to think of the movement of larger molecules that will have endocytosis paired with exocytosis to go directly across the cell. And as we're looking at the flow of our liquids through our cells, and from the blood plasma to the extracellular fluid, there are two forces at play here that are at a tug of war. We have hydrostatic forces and we have osmotic forces. We'll have blood hydrostatic forces and blood osmotic forces, and we're also going to have extracellular hydrostatic and extracellular osmotic forces. To generalize, hydrostatic forces will always push something across a membrane, push a fluid across a membrane. Osmotic forces pull a fluid across a membrane. So when we think of blood hydrostatic forces, that's going to be from the blood pressure. As the left ventricle squeezes the blood into the systemic circuit, it pressurizes our blood and pushes liquid out of the bloodstream through blood hydrostatic forces. We're also going to have osmotic pressure in our blood. Albumin is the most common protein in our blood serum, and it serves the purpose to help regulate blood osmotic pressure. If we have too little blood protein, too little albumin in our blood, we lose the ability to pull liquid back into our bloodstream with osmotic pressure. What we find, and don't pay attention to the math, I'm more interested in the big picture. Generally speaking, the arterial capillary is so strong that most of the liquid will leave the blood capillary on the arterial side. However, on the venous side of our capillary bed, osmotic pressure in our bloodstream becomes so much stronger relative to hydrostatic pressure that instead of pushing liquid out of the blood capillary, we pull fluids back in on the venous end of our blood capillary. It's of low pressure. The superior cava muscle pumps. One of the reasons that people who exercise have better blood flow is they're contracting their skeletal muscles. And as they contract their skeletal muscles, they're going to squeeze the veins, blood moving in one direction, one-way valves within the veins, known as the venous valves. And then finally, the last mechanism that draws blood back to the heart is going to be cardiac suction. As atria relax in atrial dystole, volume, back to the heart. So we have a review question. And we have 3 minutes left of class. So, 60 seconds. is a system. As we look at our flows here. A, going from the heart to an artery to back to the heart. Back to an arterial, back to another capillary bed, venual vein and heart. For C, we have heart, artery, arterial. And for D, we have arterial. It's going to be a portal system. So make sure you chat with yourselves. We're done in 10 seconds. Submit your answers if you haven't done so yet. Hush. We still have class time. Some people are still trying to learn and take advantage of their class time. If you want to be disruptive towards them, don't come to class. When we think of a portal system has two capillary beds. That's the key characteristic of a portal system. So as we're looking at our options here, the option B, where we have a capillary bed arterial to another capillary bed, is the correct answer for the portal system. And we don't have time for the other question. I had to skip. So now we're done with class. See you on Thursday.